Okay. Good evening, students. So last week, unfortunately, I sent the Zoom link, but I hope you were unable to join it. Right? It was a morning session, right? So so we are back with the unit two theory completion, right? Okay. Now I hope last topic which we discussed was lipids. Am I right? So in lipids, we completed what are lipids, what are the characteristic features of lipids, right? And then we spoke about how lipids or fats are produced, what are the types of lipids, and then we understood that what are the functions of lipids, right? So all of those were completely done, right? Last week, right? Am I right? I hope that was the last topic we discussed. I hope protein was not started last class, right? Am I right? We didn't start proteins yet, right? Hello? We didn't start proteins yet, right? Ah, okay, fine, fine. Nihla, you joined last class, right? No? No. Oh. Last class where we discussed lipids, no. You didn't join. You didn't join, right? No, last week we didn't have class. Ah, you didn't. Okay. Okay, then you too, if you haven't completed this lipid part yet, right? No issue. This is not connected with the past sections, right? This is a new part. So what you do is I'll be sending the YouTube, the recordings of this, right? I don't know whether you have watched it, so I would be uploading the recording, right? So for you too, you have, as you haven't watched those recordings, I'll send it, right? You just watch it when you are having some free time and then complete those sections, right? Okay. You okay? Not now. When you have time, just do it, right? Uh, have you completed these sections in your any other classes you are going? No? Is this the first time you are coming across this protein section? Yes, first time. For one student, it's first time it seems. Okay, then, no issue, right? So I'll send the recordings. You just free when you have free time, just watch them. No issue, right? Okay. So today's topic is proteins. Now, what is happening here? In unit number one, we understood what is biology, right? So that is also I have done a session, separate session, right? So if you are a new student who have joined today, no issue. I'll be sending the recordings, right? So you just watch them. Unit one I have completed. Unit two now. Unit two we introduced with water. After water we went to the organic molecules. Carbohydrates was done. Lipids was done last week, and today we'll be starting proteins, right? So another one topic more after proteins, it is nucleic acid. After nucleic acid is done, this organic molecule part will be done, not the unit 2. Unit 2 has some more topics. I'm saying there's another the after, no, in unit 2, wait, I'll give you a small idea, right? First of all, what you are studying. So here in unit 2 there are several topics. First topic which was discussed was about water. What is water? How water is, actually it has the structure of water, the properties of water was discussed there. Now after water we start a topic which is organic molecules, right? About organic molecules. So in these organic molecules what we studied was there are four types in our syllabus. First one was carbohydrates, right? And then we went on to lipids, fats, right? And then we now in the third topic is proteins, and the fourth topic would be nucleic acids, right? Fourth topic would be nucleic acids, right? Wait a minute. right? Nucleic acids, right? So after completing this, we can complete this second topic. After that, we have several other topics. Microscopes are there, cell biology, we have cell division, metabolism, photosynthesis, and we have cellular respiration. If we complete all those topics, we'll be completing the unit number 2, right? This is the rough idea, right? Okay, now today we'll be starting protein section, right? Okay, now have you learned about proteins before? You might have learned, right, in your O levels, right? But there you might have learned about, they have given us some sort of a minor introduction there, which means they have just said that proteins are used here, proteins have this amount of functions in human body, proteins are necessary for our diet, right? You might have learned like this. So here what we'll be learning is we'll be actually looking into the, a bit deeper into proteins, right? Okay, let's go on to the note. Proteins. Now, do you have this tute? I have shared this in the group. I don't know whether you have the tute. No? You have the tute. Oh, one student has the tute. How about the other? Do you have the tute? Oh, but still, I will share this screen, right? So you can better watch it, right? Okay. Proteins, right? Let's get into the notes. Proteins are made up of amino acids, right? Proteins are made up of amino acids. Now, proteins are some sort of polymers, biopolymers. Now, the word polymer, is it new to you all? The word polymer. The word polymer, is it new to you all? No. I hope you have learned this in your O levels. Those polymers, hydrocarbons, can you remember? Alkane, alkene, alkyne. On those sections, I hope you might have learned uh, about polymers there. Am I right? No, it's too long since I have finished all of us, so I can't remember it correctly. You recently completed it, so you might be a bit expert, right, in those, right? So there you might have learned about polymers. Simply, polymers is what? A collection of components, right? Just think a large molecule, right, which are made up of small, small molecules. So, what are the small, small molecules that make a polymer that has a common name? What is that common name? What is that name? What are the small molecules which form a large molecule or a large polymer? The small molecules which form a polymer is called, the basic units of a polymer are called. No, this is not in your A-level syllabus. It is not in your biology part, right? This is from your O-level knowledge. Monomers. Have you come across the word monomers? Now, if you can use the mic, it would be better. So I don't want to keep on watching the chat, right? If you can, it would be a great help, right? So the word monomer, is it new to y'all? No, right? No, you're a bit familiar to those words. Right? So just think, protein is a polymer, a biopolymer, and the monomer is amino acid. Is that clear? Proteins are made up of amino acids, right? Okay. 20 amino acid, 20 different amino acids are involved in the formation of protein. 20 amino acids would be involving like this. 20 amino acids will be involving like this, and they'll be forming a protein. Each would be different, it seems. 20 different amino acids. Elemental composition is carbon, hydrogen, oxygen, nitrogen, sulfur. Proteins would contain these elements in them. It seems they contain these elements. What are the elements? Carbon, hydrogen, oxygen, nitrogen, sulfur. Clear? I hope these first three statements are clear, right? Nothing hard to understand, right? Hopefully, it's complete protein within today, right? So The first three statements. Is it clear? You should be very interactive with me so I can make it bit engaging, right? So rather than making it bit boring, I'll try to make it bit engaging. If you interact with me, it is very useful for me, right? It will be helpful for me actually, right? Is these first three statements clear to you? Any doubts? Yeah, one student clear it seems. How about the other? If you find any difficulties, please let me know. If you have any connection issues or no, any other issues, is it clear for all? Clear. Very good. Fourth statement. At the center of amino acid is an asymmetric carbon atom except in glycine. Now, this is the structure how an amino acid looks like. See this, right? This is how a amino acid molecule looks like. It has a carbon atom at the center, right? The carbon has a small, there's a small symbol like, the proportionality symbol, like this, right? Like that, there's a small symbol indicating that this carbon is not just a normal carbon. This carbon has a specific name, a special name called, what is that name? What is that name? Asymmetric carbon. Asymmetric carbon, right? Okay, I'll explain what is that asymmetric carbon, right? So let's see what are the compounds that are present in a amino acid structure. Okay, now there is an amine group. Nitrogen and hydrogen are forming an amine group, right? It has an amine group. It has a hydrogen atom. And what is this called? A carboxyl group. COOH means a carboxyl group is present. And R is called an alkyl group, right? So these are the components that are present in a amino acid. So similarly, if this is one amino acid, how many such amino acids are required to form a protein? If this structure is the structure of a single amino acid, how many such amino acids are required to form a protein? 20. Very good, very good. Right? So similarly, 20 will be involved in forming a protein. But are the 20 equal to each other? Are they identical? Yes or no? Are they identical or are they unique? Are they different from each other? Are they the same amino acids? Different amino acids. Very good, very good. Right? Okay, you are clear with that, right? Okay. So what is this asymmetric carbon actually mean? Asymmetric carbon means if the four groups, now we know that carbon has a valency of 4. Am I right? This is from your overall knowledge. Carbon is present in the group number 4, right? Now you will be studying that the actual group is not group 4, it is group 40, right? In your A level syllabus, you will be learning it is not actually group 4, it is group 40, right? But for the moment, as you are coming from the O levels, I'm saying that carbon, as per your syllabus, carbon is belonging to fourth group. So it has a valency of 4. It has a valency of 4. So as it has a valency of 4, it can bind to 4 different groups. Am I right? It can bind to 4 groups. Now if you see, can you remember this structure? What is this structure called? Methane. Can you remember? So if you see that structure, methane, very good, right? So it will be looking like this. One carbon atom, right? 4 hydrogen atom, right? It will be looking like something like this, right? It will be looking something like that. So can you see there are 4 locations or 4 vacancies where carbon allocates its vacancies for 4 groups, right? So now here can you see all 4 groups are symmetrical. Symmetrical means all four groups are actually identical. He also hydrogen, he also hydrogen, he also hydrogen, he also hydrogen. So is this a chiral carbon or is this a asymmetric carbon? Means no. Why? The requirement or actually asymmetric carbon means all four groups should be different from each other. Now see here, this is carboxyl group, this is alkyl group, this is amine group, this is hydrogen atom. So all four are different, all four are unique. So can you see what actually asymmetric is meant? What does actually asymmetric means? Is that clear? Yes or no? No means no issue. Just yes, right, fine, very good. Okay. So now tell me, is methane having an asymmetric carbon? Is methane having an asymmetric carbon? Yes or no? Check and let me know. Does it have an asymmetric carbon? Just check this diagram. Does it have an asymmetric carbon? Now, if you can't understand, let me know. No issue. I won't be scolding something. No, this does not have an asymmetric carbon. Why? What is the reason? What is the reason? Because it does not have an asymmetric carbon. Same groups are attached. So same groups cannot be present. Right? Okay. Okay. But see, this part is okay. At the center of the amino acid is an asymmetric carbon. I explained what is an asymmetric carbon. Right? Except in glycine. Now in glycine, what happens is they would have similar groups, which means this alkyl group is present, right? Instead of this alkyl group, they would contain an hydrogen atom. So if an hydrogen atom is present, what happens? Are they identical or not? There are two hydrogens, right? Can you see? If this R is replaced with an H, is this carbon an asymmetric carbon? Now this is the thing. Now here there's a R, right? Now don't think that what is this R? There is no element such called that R in our syllabus, right? You might feel like that. Now here R means actually alkyl group means, now here's a hint. See, chemistry normally we use like this, right? Now there are groups like, continuously we would wait. Sometimes it will be going like a chain like this structures. So it will be going like this, it will be, it will be going on. So instead of writing like this, right, we will be simply writing in alkyl group like this. R means there would be a chain, something like a carbon skeleton, right? There would be a carbon length. So instead of that we can write an R. Now my question was, if this, so should all be attached same to be asymmetric, or if two or three are similar? I didn't get you. So should all be attached same to be symmetric. To be asymmetric, the simple idea is four groups, right? All four should be different. None of them can be, identical. None of them. Even two of them are identical. It is not asymmetric. Yes, all attachments should be non-identical. Clear, right? No doubts in that. No doubts in that, right? I hope it's clear. Right? Okay. Now my question was, just think, if I remove this R and instead of that R, what if I replace it with a hydrogen atom. If I replace it with an hydrogen atom, a hydrogen atom. Now tell me whether this structure is a asymmetric one, which means now this carbon, whether this carbon is asymmetric or not. Is that carbon asymmetric now? No. Why? What is the reason? What is the reason? Two groups are identical. What are the two groups? Hydrogen. So two hydrogens are present. For a carbon to be asymmetric, all groups attached should be non-identical to each other, which means they should be unique, right? They should be different groups. I hope that's clear. Right. Let's go to the next point. Each amino acid is composed of an amino group. This is the amino group, carboxyl group, hydrogen atom, and a variable group that is symbolized by R. This variable group is the one that will be changing from amino acid to amino acid. All others would be same. Can you see? Now this bottom structures, I'll mark it here. Now if you see, wait, right, these three, the amino group, the carboxyl group, the hydrogen atom. These three would be same for all amino acids. What is the thing that is changing in an amino acid? From amino acid to amino acid, what is changing? What is the one changing? Only the variable group, the alkyl group, right? That is the one that is changing from amino acid to amino acid, right? Okay. In the case of glycine, R is replaced with hydrogen atom. I said you that, right? I said that in glycine, this R is replaced with hydrogen, so it is not an asymmetric carbon. The R group, also called the side chain, differs with each amino acid, whereas the other groups are called backbone. Now see this. The green part that I have mentioned is called actually the backbone. What is it called? It is called the backbone. Wait a minute. Right? This is the green part. This is called, it is the one that is called the backbone. This R group is called, this R. That is the one that is called this one. This R is called side chain. So these three components which are constant from amino acid to amino acid is called backbone. This the variable group, the R group which is changing, that is the one that is called side chain. Now listen carefully and answer this. From amino acid to amino acid, is the backbone changing or the side chain changing? Does the backbone change or the side chain? Which is the one changing? Side chain. Very good. Right, right. Backbone remains constant. Very good. You have got the point. Right? You have understood that. Right? Guys, give me 1 minute because you can't see the screen right now. Wait, wait. Right. Now can you see the screen? Can you see the screen? Students, can you see the screen? No, there's a small issue. Now, is it fine? Can you see the screen? Yes, right. Okay. Right. Okay. So let's go to the next point. Amino acids may have one or more carboxyl groups and amino groups. Now just check this diagram and let me know. Can an amino acid have more than one carboxyl group and one amino group? Now you might say, no, sir. If you see there are four groups, only one carboxyl group and one amino group can be present, right? How more? If you ask how, this variable group that is varying, right? So that can also have a structure like this. Now the carboxyl group, we can, you have got the point now. Very good, very good. Now you have got it. What is actually now, without my explanation, you can understand it now. See here, one student has said it that this R group, instead of that, if a carboxyl group is present, if a carboxyl group is present, or an amine group is present, uh, now we can say that there would be more than one amine group or more than one carboxyl group. Right? Okay. That's clear, right? I hope that's clear for all. Is that clear for all? That point? Now try to understand each and every point, right? Right. Now, by the way, uh, are both of you in the group of mine? Which means now the group where I share the Zoom link. I hope you are in that group, right? Both of you. Now, both of you, did you get the uh, essays which I sent? Now we have planned of sending some essays, right? So just to practice you, I would be sending some MCQs randomly, right? I won't be giving huge pressure on you because you have to manage other classes also, right? I'll be sending gently. You got it. Very good. I'll be sending gently MCQ paper, MCQ questions, right? Essays would be sent. You just write it and take a photograph and send me. I'll correct where, I'll correct how, what are the mistakes that have you have done, right? And I'll make you aware how to write it, right? So that's one of the help I can do to you, right? Now if you are attending physical, I could give it, correct it physical and give you, but as you are joining online, I have to ensure that you are doing the works, right? So make sure to do the papers or essays that I've sent within the span of time I have given. If I give a deadline, do it within that period and just send it to me, right? Okay, right. Let's go to the other point. Amino group, amino group has alkaline nature and carboxyl group has acidic nature. What is this alkaline and acidic means? Alkaline means, alkaline means no idea what is alkaline meant by. I'm sure that you are a bit familiar with acidic. What is meant by acidic? Acidic means a lower pH. Yes, acidic means a lower pH. Alkaline means basic nature or higher pH than the neutral, right? So amino group as these amino acids. Now simply this is simple. Don't make it complex, right? I'll explain it by statement. No hurries. Right? Within this today's session, you will be completely thorough with this protein, right? Don't worry. As you are coming across these words, right, for the first time, don't get panic. Let's do it calmly, right, in composed manner, right? Okay. Now this is an amino acid, this structure, right? As this structure has an amino group and a carboxyl group, if you see that amino group is present and this amino groups gives what nature? The amino as amino groups are present, this amino acid gets a basic nature. Is that clear? As amino groups are present, the amino acid gets a basic nature. Yes or no? Is that clear? One student has said it's clear. Simple, simply like a school classroom, right? If there are one bad boy or bad student, just because of his atrocious behavior, sometime a group of students would be blamed as bad students just due to that one student. This happens, right? No, that's a common scenario like that. As amine group is present, amine group is basic, it has a high pH, right? But just because amine group is present, amino acid also gets an basic nature. Is that clear for all? All students, please make sure to give me a response. If it's clear, yes. No means no. I'll explain. Yes, clear. Very good. Okay. This carboxyl group is naturally acidic in nature, which means it has a lower pH. So, as carboxyl group is acidic in nature, just because amino acid has carboxyl group, amino acid gets an acidic nature as well. Now it got a basic nature first because of amino group. Now it gets an acidic nature due to carboxyl group. Now what is the type of nature that amino acid has? It has both acidic nature and basic nature. Now you can just give an hint or just give an guess how this amino acid would look like. What is its actual nature looks like? It has both acidic and basic nature, right? Okay, so that property of having both acidic nature and basic nature is called amphotericness, or it is called his amphoteric nature. See, when both characteristics are found in one molecule, they are known as amphoteric molecule. Is amino acid an amphoteric molecule? Yes or no? Yes. Amphoteric means, amphoteric means, amphoteric means having both acidic and basic, or acidic and alkaline nature. Very good. Both acidic and basic should be present. If that is the case, they are called amphoteric molecules. Now, just go through this part, the first page, and let me know if you have any doubts. If you have no doubts, let's move to the next part. So this part, this complete page, just go through it. Yeah, alkaline means basic, right? Alkaline means basic. Basic means the pH, right? In the pH, more than 7 means basic nature or alkaline nature. Less than 7 means acidic nature. That basic, right? Clear? Is that clear? For the one who asked the question. Clear, right? Very good. Just check now this complete page. If you both are clear with this part, both of you drop a message saying clear. Then I'll move on, right? Just check it. Is it fine if those points are included in the X? Which point are you asking? These points, which they are given here, these bullet points. Yeah, this should be included. Now, if they ask a question related to proteins, you should include them depending on the question they have asked. If they ask, explain briefly the characteristic features of proteins, you should include all of them, and you should draw the diagram as well. For drawing the diagram, also they'll be giving marks, right? For drawing, they'll be giving a mark. For labeling, they'll be giving a mark, right? Okay, so don't worry. If you haven't tried any essays yet, no issue, right? I'll be starting, gently. I'll be giving you one essay per week or two essays per week, not more than two, one or two essays per week, and randomly for 2 week, once in a 2 week, I'll be sending a paper also, right? Do it and send it to me. I'll correct it and I'll give, right? uh, Explanation, they won't be exactly expecting those explanation, because mostly they'll be giving marks for the terms that they are given in the resource book. They won't be giving marks for your own writing, which means now if you include something that is not included here based on your knowledge, even if it is correct, they won't be giving marks, because they expect only the terms that are given in this resource book. Clear? Okay, right. So even if you want to explain something, you can explain within the range of resource book. Now, if you see here, you can say that amino acids are amphoteric molecules, and you can explain what is amphoteric molecules. That is fine. Why? Because they have given it in the resource book. Now, if you go on explaining further, what now? If you example, just think here. Now here they haven't said what is asymmetric carbon. This is the resource book note. Now see here, have they mentioned what is asymmetric carbon here? Have they explained what is asymmetric carbon means? Just check. No. Now, if you write that asymmetric proteins have normally the amino acids in the proteins have asymmetric carbon. Asymmetric carbon means having four different groups. If you keep on explaining those sections, even that is correct, they won't be giving marks because it is not included in the resource book. Examiners, they will be expecting only what are the contents that are present in the resource book. Clear? Is it clear? Yes. Okay. Okay, then just go through this part, and if it's clear, drop a message saying clear. Okay, one student is fine. How about the other? Is it clear for you, Nihla? This first page. Now, even if you have any difficulty with respect to one word, no issue. Drop a message. I'll explain it again for you. Just check this part. If you have any doubts, drop a message. No doubts means drop a message saying, "Sir, clear, sir." If you respond only, I can move on. Right? Is this section clear? Is it clear? Is it clear for all? Nihila, are you there? Is it clear for you? Yes, very good. Okay. Then we can move to the next part. Right. Therefore, amino acids are amphoteric molecules. We explain why they are amphoteric. Right. Two amino acids undergo condensation reaction. Condensation reaction means, now if you have the resource book, I hope you both have the resource book. If you have the resource book, under that note part, right, just underline that condensation and above it write condensation reaction means water removing reaction. Okay. No, sir. Nihila, why have you mentioned no? You don't have the resource book. You don't have the resource book, or you have? You don't have. Okay. No issue. Now you haven't taken the tube print out, right? Did you get a printout? Did you get a printout or not? You have the then no issue. If you have the print copy of this tube, then you can you just take this page in the tute and underline this condensation reaction part and above it write water removing reaction. Okay. Okay, right. After writing it, let me know, then we can go further. Right, condensation reaction means water removing reaction. Right. Let's see how that reaction is formed. Oh, that condensation reaction takes place. Now this is the thing. We know that how many amino acids are involving in forming a protein. How many amino acids are involving in the formation of a protein? They gave a number. How many? Guys? How many? 20. Very good, very good. 20. Right? Now this is one amino acid. It should be linked with another amino acid, right? If it is linked only, just think of a chain of pebbles like that. It should be linked, right? If it is linked only, it can form a chain of amino acid to form a protein. Clear? First, it should link, right? Just by having a single amino acid, we can't form a complete protein. We should link them with, or we should join them with other amino acids, 20 amino acids to form a protein. Is that clear? Yes or no? Yes or no? Yes, very good. Now see, this is an amino acid. This is an amino acid. Just check how they are linking. Now, this is the carboxyl group. Now, carboxyl group we studied it as COOH, right? Now this is the expanded version. There is a double bond O and a single bond OH group. Are you fine with that? That structure? Is it any confusing with that structure? You want me to explain that structure? Fine, right? How about the other? Is it fine or should you want any explanation? This. Now they studied that amino acids have an amine group and a carboxyl group. Carboxyl group means here see they said that it is looking like the double bond O COOH, right? Now instead of writing that, they have given it in the expanded form. See, there's a double bond O and OH group. Is it clear? Fine, very good. Now here amine group means NH2. So there are two hydrogens and NH. This is the expanded form. Now see, there's a reaction that is taking place. This red color OH and this red color H are being removed as a water molecule. Water molecule has how many hydrogen? Water molecule has how many hydrogen? H2O. How many hydrogen? How many hydrogen? Two hydrogen, right? So here see, one hydrogen from amino group of one amino acid and one hydrogen from the carboxyl group of amino acid. Can you see? It is not removed from the same group. It is removed from the amino group of one amino acid and the carboxyl group of one amino acid. Those groups are removed, and just think like this. Now see here, these two are removed, right? And that these two are joined. See here, by removing that those two groups, they have made a bond. Can you see? Linking two amino acids. Can you see? By removing the OH group from carboxyl group and H from amino group, they removed that water molecule and they joined those two groups by a bond. Can you see that red color bond? See here. Is it clear for you? These two are connected after the removal of water. Is it clear? No, do you find any difficulties in understanding? If you respond only, I can know what you're going through. Is there like kind of electrostatic attraction between those to join them exactly? If you ask electrostatically, rather than going on to electrostatic, now electrostatic means it is taking place in ionic bonds, right? Electrostatic attractions takes place in ionic bonds. Am I right? Am I right? Sodium chloride means sodium ions and chloride ions. There's an electrostatic attractions that are taking place. Here, if you see, carboxyl group is an acidic group, amino group is a basic group, acidic group and basic group. Just think this reaction HCl, right? HCl and sodium hydroxide. HCl plus sodium hydroxide produces a sodium chloride plus water. Can you see at the end of an acid group and a basic group, water is produced? Neutralization reaction. Is that clear? That was, yeah, you have studied that in your O levels. You're familiar with that reaction, right? Sodium HCl reacting with sodium hydroxide, right? So at the end, water is removed. Here also, at the end, water is removed. Why? Because this is also some sort of neutralization reaction. Why? Because here an acidic group joins with an basic group. At the end, water is removed, right? So as this reaction removes water, any reaction that removes water is called what reaction? Any reaction that removes water is called what type of reaction? What type of reaction? Condensation reaction. Very, very good. Right? Is that clear? Is it clear now? Right. Right. Now, can you see after removing that water molecule, both are very good, clear, right? Both carboxyl group of one amino acid, right? Carboxyl group of one amino acid and the amino group of other amino acid are joined by a red color bond. Can you see? Now, this is like something like this. Have you seen vehicle business? Have you seen vehicle businesses? Just no. Rather than just going on with the theory, I'll give it just a practical example. You have seen vehicle businesses. During vehicle businesses, there are brokers. There are brokers who contact them, and they are the ones who introduce the buyer to the seller. Am I right? Yes or no? Yes. Now, just think practically, why does the broker want to put such an effort bringing the buyer and the seller to a one place and make them go into an agreement and sell the product? Why should a broker make such an effort? Why? What is the only reason? To obtain a contract? Rather than using the word contract, I want a specific one. Why does a broker do such a thing? You know the word broker, right? Do you know the word broker? What does it mean? You know it, right? Normally you would say a broker. Broker means not a broking something like that. Broker means who contact someone and for some sort of work, who contacts someone and brings. Now, if you see for a vehicle business, who contacts some seller or a buyer is called a vehicle broker, right? So why does he do such an effort? What is his only motive by doing such an effort? Now, just think, this is his buyer, this is his seller. Now, he's bringing those two guys. Why? He want to sell that product, but it is not his product. Then why he is making that effort? Now, if something is not your product, why should you make such an effort? For the profit. Rather than using, no, profit is good. For instead of telling that, we can say what? To get a commission, right? Am I right? Normally brokers use the word commission, right? Okay. So just think, both the buyer and the seller are being known by a common person, who? The broker. The broker knows the seller and the buyer, right? So at the end of the business, both of them, in a sense of thanking him, they would give us some sort of money. But at the end, the broker would get from the buyer also some amount of portion or a commission, and the seller also he would get a commission. So totally, what he will do, he'll take the commissions and he will be making them interact or make them contact and do all other activities. His work is until he makes them meet, right? After making them meet, he won't be going further into their business. Am I right? Yes. Now here also the same scenario. What happens here? Just think, this reaction is the broker. The reaction takes OH from one amino acid. Just think OH as a commission from this and takes H from this. And totally, what is his total commission? What is his total commission? OH from one amino acid, H from one amino acid. What is his total commission? Guys? H2O, very good. Right? Can you see? H2O is his total commission. And what does he make? He is allowing them to come into contact. They will be making them interact. And he forms this bond and goes. Right? He forms a bond between those two, buyer and seller, and he goes. Can you just connect it with a practical scenario? Is it relatable? Got right, very good. Now, that bond that is formed between two amino acid. Now, after this bond is formed, if someone looks at this structure, will he say that this is two amino acid? Can we come to that conclusion? Can we see? Have they mentioned that there are two amino acid? No, we can't say, right? So this looks like a complete structure. So, but actually, we know as bio students now we can say that here this contains how many amino acid? This final structure. This contain how many amino acids? How many amino acids are present? Guys, how many amino acids are present here? Just give a try, no issue. If it is wrong, no issue. This is the first time you're coming across this, right? Just express your idea. As bio students, when we look at this structure, the circled part, how many amino acids are present? Two, very good. Two amino acids are present. Why? See here, one from here and the other from here, right? Can you see? But so the bond that joins two amino acid is called what type of bond? Peptide bond. Now can you see there are two amino acid. Between those two amino acid, the bond that connects amino acids are called this black color. What is this bond called? What is that bond called? What is that bond called? Now, if you can use the mic, is it's easy, right? Because rather than keep on typing it, if you can use the type, I mean, use the mic, right? A peptide bond, very good, right? Now, can you see? Can you clearly understand what is this process going on? So this process takes place just to make a peptide bond to join two amino acid. So during the joining of two amino acid, what is lost? What is lost? Think carefully and reply. When two amino acids are joined, what is lost at the end? What is lost? Now just think this as a business. What is lost for the buyer and seller at the end of the business? H and OH as separate, so products, right? Rather than H and OH, what is the final product? It is lost. Water. Am I right? Yeah, you are more precise saying that hydrogen and OH. Very good. But at the end, if you see as a total water, if you see as a total business, at the end, what is lost? Money. It is not as money from A and money from B. Total, it is money that is lost. So here, if you see in this reaction, total what is lost? Water is lost. A reaction that loses water, that removes water is called what type of reaction? What type of reaction? A reaction which removes water. Very good. Condensation reaction. So just see this diagram. I hope this is clear to you, right? Okay, let's move to the other part, right? Protein is composed of one or more polypeptide chains which are composed of amino acid. Now, when amino acid, now see here how many amino acids are present in this circle diagram. You first responded. How many amino acids are present here? No, I'm saying here. In this circle diagram, how many amino acids are present? Two. Very good. So, this is for two. Now, here, with this also, there can be another linking that can take place. Here, another linking. So, it will be continuously, there would be created and they will be forming a complete chain. Can you relate it? A chain of amino acids. Is that clear? Is that clear? Yes. That chain of amino acid is called a polypeptide chain. Can you understand that? What is meant by a polypeptide chain? A polypeptide chain means a chain of amino acids. Can you understand? They haven't given a number. If there are 20 amino acid, it is a protein. But there can be shorter chains, right? If there are 3 amino acids, it is called a polypeptide chain. If there are 4, it is a polypeptide chain. 5, 5 amino acid means it is a polypeptide chain. Can you understand? So in a protein, there can be one polypeptide or more polypeptide chains. Can you understand that? Relation to glycosidic bond. No, glycosidic bond was formed during uh, carbohydrates, right? It is a bond that is formed during, yeah, like peptide bond in proteins. Glycosidic bond was formed during during the disaccharide formation. But what is common for both the bonds? What is common for both glycosidic and peptide bond? The condensation reaction. At the end of both glycoside bond formation and peptide bond formation, water is removed. They are also similar pattern. But what is the only difference? Here, amine group and carboxyl group, they are two OH groups, right? Is that clear? Is that clear? Yes. Now, is this first point clear to you? Protein is composed of one or more polypeptide chains. I am sure you might get confused with this. Now, this is the thing. Proteins are made up of how many amino acids? 20 amino acids, right? So, if 20 amino acids are involving in forming a protein, if there's this thing, there is a 20 amino acid collection of amino acid chain. So, this is a polypeptide chain. This is the polypeptide chain. Listen carefully. This is the polypeptide chain. Here, only one polypeptide chain has involved in the protein formation, right? Just think there is a polypeptide chain like this, right? Here there is 5 amino acid, right? Wait a minute. Here, how many amino acids? So here 5. Here, how many amino acids are present? These circles are the amino acids. Just think. Here, how many amino acids are present? 10. Here, how many? The third one. Here, how many amino acids are present? 5. So, what I'm trying to convey is the collection of amino acid or the chain of amino acid. This is called a polypeptide chain, right? So, in a protein, how many amino acids should be present? 20 amino acids should be present. Here also, this these three can form a protein, but they have three polypeptide chains. Can you see? Polypeptide chains, three polypeptide chains. So there are multiple options. Now just think the main requirement is 20 amino acids should be present, right? 20 amino acids can be present, but it can have one or more polypeptide chain, which means it can be a set of 20 or it can be clusters as 5, 10, 15, 20, right? It can be clusters also. It can be a complete 20 containing chain or it can be a small clusters also. If it contains a complete chain of 20, it is called as one polypeptide chain. If it contains multiple polypeptide chains like this, we say there are more polypeptide chains. Is that clear? This is a very difficult part to understand. You want me to explain it again? I am ready. If you want to explain, I'll explain it again. Guys, fine. You got it. Clear, right? Don't get confused. Now just think like this. Now a classroom is present. Now what they say is, now there is a sir, right? There is a sir. Here now just think your school is hiring an external sir. He says that maximum I'll teach only if there are 20 students. All 20 should be present. If only 20 students are present, I will be teaching. Less than that, I won't be teaching. Right? You are hiring a very busy sir. So he's coming, and the hall they give is an auditorium. If they give an auditorium, just think it practically. All 20 students will be sitting in the front auditorium, one row. They are sitting in a one row, right? If it is an auditorium, huge auditorium, they can sit in a one row. Am I right? They can sit in one row. Is it clear? Yes. No, right? Okay, right? Right. Now they are saying auditorium is not available. You can do it in class. If you are doing it in class, what should we do? They are breaking the student. Now classes would be small, right? You can't keep 20 students at a row. So we'll be breaking students into small parts. We will be front row will be 5 students, then 5 students, then 5 students, then 5 students. Right? Now just think practically. If an auditorium is present, how many students can be present in a row? In a single row, how many students can be present? Guys, no, please try to respond. No, I'm saying auditorium. If it is an auditorium in a single row, how many students can be present? If it is a large auditorium, 20. So if it is in the class, how many can be present in a single row? How many students can be present? It can be four, it can be five, it can be maximum. Just let's say five, right? It can be four or five. But think carefully and tell me whether the sir will be teaching. You know, he'll be just can you understand? He'll be teaching in both scenario, even if it is in auditorium or if it is in the classroom. Why? Why? What was his main requirement? Was it the class? Was it the class his requirement? His demand. What was his demand? I'm trying to make it simple for you, right? What was his demand? Teaching, and he should have 20 students. That was his demand. He said that I'm very busy. If there are 20 students or more, I'll come. Minimum 20 should be present. That was his demand. Now here in this scenario, in both scenario, 20 students are present. But the difference is what? What is the difference? In both scenario, 20 students are present. But what is the difference? What is the difference? Space and arrangement. Right? In auditorium, 20 were present at a collection. They'll be as a complete row. Now they have been broken into the. Now just can you think it practically and connect this with this knowledge? In auditorium means 20 students like a complete amino acid chain. So how many polypeptides are present if it is auditorium? Just think the students as amino acids, right? If it is in auditorium, how many polypeptide chains are present? Now, if you are clear with this, you would answer. If it is in auditorium, how many polypeptide chains are present? One. Very good. Very good. You have got it. Right? If it is in the class, how many polypeptide chains are present? If you take as 4 students in a row, several chains broken. Just tell me, if there are 4 students per row, how many polypeptide chains are present? 5. Very good. You have got that. Can you understand? But at the end, protein would be formed, which means the class would be conducted. Just think, protein formation means class conducting. In both the scenarios, protein would be formed, but in different. Now see, the first scenario, in the auditorium also the class would be conducted, but they are as a single row. The students will be arranged. If it is the class, also the students will be students will be taught this. The teacher will be conducting the class, but they'll be in different rows. But at the end, what is the main the motive? Teaching takes place, right? So similarly here also, protein would be formed. It can be formed as a one polypeptide chain like in the auditorium, or as multiple polypeptide chains like in the classroom. Is that clear? Now I use this example to make it easy. If this is hard to be understood, no issue. Now some might feel like that, like this, right? Sir, you taught it first with these balls. I was clear with that. I can't understand this auditorium scenario. If it's that's fine, right? I want to understand this concept. I don't want that auditorium part to be remembered. If you understood with this concept, that's fine. Don't try to understand the example which I'm trying to explain, right? Is that clear? This point part, is it clear? Yes, one student has said it is clear. How about Nehla? Is it clear for you? Don't worry if if you're not clear. Until you are clear with this, I would teach, right? So better mention it. Sure, right? No doubt for both of you. Don't move on with doubts. It would get accumulated, right? Clear, right? Now I'm trusting your words. So you have said that it's clear, right? Very good. Right now, this is the thing happens. Now, what is it formed? Now, amino acid is formed. Amino acid chain. Just think the amino acid chain is formed. This amino acid chain, as it forms, when amino acid chain is a form, just think a chain of amino acid. A chain of amino acid has another name. What is it? What is it? A chain of amino acids has another name that can be called polypeptide chain. Why they have used the word polypeptide? If you ask me why say it is polypeptide, because if you see between two amino acid, one peptide bond is present. Between three, two peptide bonds. So multiple amino acid means there will be multiple peptide bonds. So as there are multiple peptide bonds that are formed, we say that it is a polypeptide chain. Is that clear? Now this is a polypeptide chain. This complete chain. See, this one circle is an amino acid. So this is a complete polypeptide chain because there are multiple peptide bonds between each of them. See, if you see this black one as the peptide bond, these are the peptide bonds that are present between two, right here like this. So there are multiple peptide bonds. As there are multiple peptide bonds, we say this complete structure as a polypeptide chain. This is complete a polypeptide chain. Clear, right? Right. I think it is clear. Right. This polypeptide chain, as it is formed, it won't be functional. The protein should be functional to be used in human body or in plant body. For a protein to function, it should be functional. Can you get the point? Unless it's functional, it cannot be used in various functions. Now, like a normal country citizen, he should pass the age of 18 to be used as a labor. He should be mature. Is that clear? A citizen, a student, should pass the age of 18. Unless he's past the age of 18, he cannot be given a labor work. He cannot marry. Right? There are several procedures. The limit is he should pass the age of 18 and he should be mature enough, right? So similarly here also, the polypeptides, just as they are made a long chain, they cannot be used as a protein unless they are functional. Is that clear? Is that clear? Is that clear? Yes, clear, right? Okay. So how can we make this functional? This polypeptides functional? We have four forms of protein. We have four forms of protein, which explains how proteins become functional from one, now like our school journey. So from grade 1 to grade 12, 13, how our life goes on to make a advanced level student. So what factors affect the function? What factors affect the function means normally we'll be learning this under enzyme section, right? Normally temperature, right? pH, these are the ones that affect, right? But here under proteins they haven't given those sections, right? So they won't be asking such questions. But under enzymes they have said that there are factors that affect the enzyme activity, and they have given, because enzymes are also proteins, right? So they have given that temperature, enzyme, and they have given two others also, right? So they will be learning under those sections, but they haven't given generally what affects the function. But there is another section, I'll explain it, right? So for the time being, let's go on to these parts. Is it okay? Okay. Now, like in our school journey, where there are several stages. First, we'll be in kindergarten, then we'll be going on to the junior school, and then to the middle school, and we'll be going to the secondary school, which means the higher school. And then we'll be going to the college universities, and they'll be like, there are stages, right? There are various stages. Like that, as soon as an amino acid chain is formed, it will be passing through several structures, which means it will first be a primary structure, and then it will be developing into a what type of structure? Secondary structure, and then it will be developed into a tertiary structure, and then go to the quaternary structure. So what are these? These are the different zones in which a amino acid chain is being present. It will be passing them to become a function. So when it passes this quaternary structure, it will be functional. Is that clear? Similar to your A level journey. When you pass out your advanced level, you are a mature student because you have your age is also 18 plus, right? So you can apply for any jobs and you are eligible. Is that clear? Is that clear? Clear, right? Very good. Okay, let's go on to the primary structure. Clear, right? Very good. Primary structure. What is this primary structure? So let's see. The unique sequence of linearly arranged amino acids linked by peptide bonds is the primary structure of protein. Is it hard to understand? Just read that point and see. With the knowledge we have studied so far, just read that and see. Do you find any difficulties in understanding that? So what they have said primary structure of a protein means the unique sequence of linearly arranged amino acid, which means just a polypeptide chain. Now, as soon as it is formed a complete chain, how it looks like? Now it will be looking like this, right? Right, it will be looking like a chain, right? It will be looking like a chain like this. So this complete chain, as soon as it is formed, is called a primary structure. That's it, right? When it is formed, complete chain, when it is as soon as it is formed, it is called the primary structure of protein. Can you understand? The amino acid sequence, as soon as it is formed, it is called as a primary structure of protein. Is that clear? Yes. How about the other? Is it clear? Is it clear for all? Don't mark. Don't just drop a message saying yes. It's clear, sir. Just for the sake of mentioning, if it's really clear, mention it's clear. If you have any doubt, no issue. Just point it out. Right. I hope it's clear. Right. Primary structure is clear. Right. Clear. Right. Okay. Now after primary structure, which means from that linear sequence of amino acid, it is moving into the secondary structure. Got. Secondary structure. What happens in the secondary structure? Let's see. The primary structure of a single polypeptide chain coils and folds. Just think that complete sequence, it coils and forms. Coils and folds as a result of intramolecular hydrogen bonds between the oxygen atom and the hydrogen atom attached to the nitrogen atom of the same polypeptide chain's backbone to form secondary structure. So let's see the structure. Now, if you see, just see this structure they have given. From this, you might get an idea. Now, if you see here, what is this? This C double bond O. This C double bond O is the, what is it? Carboxyl group, right? Now, if you see, this is an oxygen atom, right? This oxygen atom is being attached with the hydrogen atom of the amino group. Now, see here, I'll go back to the initial part, right? Wait. It's a bit slow. Wait. I'll hope it's this. Not this one. Other one. All right, here. Now, if you see, this is an amino acid, right? Now, this is an amino acid chain. Just think, this is an amino acid chain. Why? Because there are two amino acids. So, due to that coiling and folding between them, I just think two balls that are joined with, they're joined like this. And now there are multiple balls, amino acids. But for the explanation, I'm saying there are two like this in the which they have given the diagram. They are coiling and folding in such a manner that this hydrogen atom forms bond with this oxygen atom. Can you see? Can you see? It forms in such a manner, it coils. The hydrogen atom attached to the nitrogen atom forms bonds with the oxygen. Can you see? It forms with the oxygen. Which oxygen? The oxygen attached to the carboxyl group, the carbon atom. Can you understand? Yes, for one student it's clear. How about the other? How about the other? Nehla, is it clear? So this bond that is formed between the hydrogen, clear, right? Very good. This bond that is formed between the hydrogen atom of the nitrogen atom and the oxygen atom is called hydrogen bond. Now you know from the overall knowledge, hydrogen bonds are formed between highly electronegative elements, right? It is formed between oxygen, it is formed with fluorine, right? So this is hydrogen bond, right? Hydrogen bonds are formed here. It is formed with oxygen, right? Okay, right. Now they have used another word. Now can you tell, just looking this diagram, let me know uh, between what component and what component is the bond formed. Is it with the backbone and the side chain, or is it with the side chain and side chain, or is it with backbone and backbone? Now if you answer this, you have got the point accurately. Just see, by seeing the diagram, tell me the bond is formed between backbone and backbone, side chain and side chain, or backbone and side chain. Three options. What is the option? What is the actual scenario happening? Look it carefully. Think carefully and answer. Where have the bond formed? Is it formed between the two backbones? Is it formed between the backbone and side chain, or side chain and side chain? It's a really difficult question. Backbone and side chain circles, there is two identical edges. Using backbone and side chain. Now, wait. Now, here, if you see, I'll separate it like this. Now, this is one amino acid, this is one amino acid. I'll separate like this. So, here, one amino acid is present. I'll mark number one. Here it is two. Right? So, in this amino acid, what belongs to backbone? What belongs to backbone? What are the components belonging to backbone? Name one by one. What are the components in an amino acid belonging to backbone? See here. What are the components present in a backbone? By seeing this, at least drop a message. Carboxyl group, hydrogen atom, amino group. What is present is a side chain, alkyl group, R group, or variable group. Right? Very good. Right? So in backbone, how many components are present? Number of components or number of groups? No, backbone there are three. So you should be careful. See here, amino group, carboxyl group, hydrogen atom also. Is it clear? See the box I have drawn? Is it clear? Yes, three. Very good. So R belongs to the side chain, which is the alkyl group. Now, with this knowledge, tell me hydrogen bond is formed between what groups? Two backbones. Uh, your idea is correct, but there's a small mistake. In one amino acid, how many backbones are present? In one amino acid, how many backbones are present? No, no. I'm not asking how many components in backbone. I'm asking how many backbone. No, 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 wait. Now this is the thing. Now this is an amino acid. In this amino acid, how many backbones are present? Now don't think like this. Some go to this misconception saying that COH is a backbone, H is a backbone, H2N is a backbone. No, no, no. These three addition forms one backbone. Can you understand? All these three, when these are present, including this asymmetric carbon, this would give these four components would give us the backbone. So initially I said, what are the components in a backbone? So you should be clear with the question. So here I asked, in an amino acid, how many backbones are present? Now tell me, how many backbones? How many backbones are present? Yeah, four groups are present. There are four groups, right? Four groups means there are three groups and a carbon atom. So totally four components are present. All these four components, when they are present, these complete four components gives only one backbone. So just think here, these four have a common one box, right? So it is completely called backbone, one backbone. Can you understand? So how many side chains are present? How many side chains? One. How many backbones? How many backbones? Just give me the number. One. How many components in a backbone? Four. Very good. Is that clear? Is that clear? Is that clear for all? Nihala, is it clear? Is it clear? Nihala? Just drop a message if it's right, clear, right? Very good. Okay. Now let's go to that bond formation part. Now check it and tell me how where has the bond formed between what structures? Backbone, side chain, those. Now you might have a clear idea. It is formed between the what? Carboxyl group and the amino group. Very good. But I want the word backbone side chain. With those, try to tell me something like that. So in this group, in this now, what is the side chain in this amino acid? What is the side chain in this amino acid? What is the group that belongs to side chain? This R belongs to side chain. Am I right? No, not amino group. Not amino group. Don't confuse with that. For side chain, see here. I'll again go back to the diagram. See here. Side chain is always R. All others except for that side chain is called backbone. So amino group belongs to backbone, R group, right? So except for this R, all others are called what group or what? What is it called? Except for this R, all others are called by a common name called, what is that common name called? Backbone. Now tell me, where has the bond formed? Now I'll tell it, right? I'll tell where it is formed. Check it carefully whether I'm saying correctly, right? So the hydrogen bond, listen carefully, see this diagram, and listen carefully what I'm saying, right? The hydrogen bond, the yellow color one, the hydrogen bond is formed between the hydrogen atom of the nitrogen atom of the amine group and the oxygen atom of the carboxyl group of the same backbone. Is it correct or wrong? Correct or wrong? Students, same backbone, sir. Yes. Can you see? So here you should not say two backbones. Why? Because it is taking place within one amino acid, not between two. It is not taking place between these two. It is taking place within the same amino acid. So we should use the word same backbone. So, but two components of the backbone here. Yes, two components of the backbone, right? Two components of the backbone. So that is the thing we are saying it as hydrogen atom of the nitrogen atom of the amino group and the oxygen atom of the carboxyl group of the same backbone. We should explain with respect to the atoms, not with between the groups. Why? Why shouldn't we use the groups? Now you might have questions, so we can say that directly as between the amino group and carboxyl group, right? You might have a question saying, sir, instead of using such a long explanation, hydrogen atom of nitrogen atom, those we can say directly as hydrogen bond is formed between amino group and carboxyl group of the same backbone, right? Do you have that doubt? Do you have that doubt? Yes. But why can't we say the hydrogen bond is formed between amino group and carboxyl group? Why can't we say like that? Why? Why means, now can you see here carboxyl group? Is there any carboxyl group here now? Is there any carboxyl group here now? No. Why? Because during the condensation reaction in the carboxyl group, OH group was removed. If an OH group is removed, is it now a carboxyl group? Eh? Is it now a carboxyl group? No. Right. If this is with respect to this. Now, if you come to this amino acid here, here also we can't say, now here we can't say it as carboxyl group. Here we can't say it as amino group. Why? Because in the amino group, one hydrogen is removed. Can you see there is a problem with respect to the, if you use in groups, right? So instead of saying it like that, see here how they have said nicely. Yeah. Now see with those ideas I'll explain. See here the primary structure of a single polypeptide chain coils and folds due to intramolecular hydro intra means within the molecule intramolecular hydrogen bonds between the oxygen atom and the hydrogen atom attached to the nitrogen atom of the same polypeptide chain's backbone. Is that clear? So there are instances where this can form. So there are two ideas. One is it forms hydrogen bonds between the same backbone. Or another thing is we can say like this also. Don't confuse, right? Just patiently listen to it. Don't get hurry. We should complete this. I should try to get these into my mind. No, don't put a pressure like that, right? So let's see. Now this is the thing. Now if there is any further polypeptide that is formed, can you see uh, the backbone? The backbone is looking in this direction. Exclude this R part, right? So the top part becomes the backbone. Then use that. You is it clear the first part? Now, if I explain this, it would bit confused. It would be surely confusing for you. Did you get the first explanation part, which I explained just now, few minutes ago? Yeah. So between the same backbone, yeah, between the same backbone of polypeptide chain, right? Is it clear? Is it clear? So due to this hydrogen bond, it will get a structure like this. Now see here, the hydrogen bonds, how they are formed, right? Between the polypeptide chains like this. So don't confuse them. Just what you should do is you continuously start, reading these how they are formed. Right? When you continuously get after seeing, when you are starting to read these, what it happens is you gently start to remember this. You won't be memorizing it. You gently just see you in the resource book. Just see this diagram also. In this diagram, it is formed between the adjacent part. Can you see? So now don't confuse like this. Sometimes you might get confused like this. Now it is not a must to form in the same amino acid. I'll explain that also. Now this is I said that this is as the backbone, right? I said this as the backbone. and the bond was formed like this. That is one instance, right? Now tell me, can this form with another oxygen that is present in the top region? There's another oxygen like carboxyl amino acid up here. Just think there's an amino acid here like this, right? And there's an amino acid, and the oxygen atom is being projected like this. Just think the oxygen atom is projected like this, right? Can it now react? Now this is the oxygen. Can this hydrogen now react with the oxygen? Means yes. Why means it is acting with the same now same backbone means don't try to think as this backbone within one backbone. Same backbone means the common backbone. The backbone is common for all, right? So I'm speaking about that backbone, same backbone. Did you confuse it with the within the same backbone? It is not like that, huh? Now same backbone means the backbone does not change from amino acid to amino acid, right? Is that clear? The backbone does not change from amino acid to amino acid. Is that clear? Is that clear? Yes. So this hydrogen can react with this oxygen. It can react with the other oxygen in this side. It can react with this oxygen. It's fine. Because why? Because all have the same backbone. Same backbone means the backbone does not change. This amino group, it is common for all. This H group, it is common for all. This CO double bond group, it is common for all. Is it clear? Is it clear for you? So I'm again repeating, don't confuse it with, no, don't think like that. The bond is formed within its own amino acid. No, if it is formed within the own amino acid, see like this. If you see this diagram, see here, this is another amino acid, this is another amino acid. The hydrogen bond, the dot dot bond, see here, the dot dot bond is formed between two amino acids. Can you see? But both of them have a same backbone. Is that clear? Is that clear? Yes. So at the end, right? So after that, after these bonds are taking place, the primary structure will be developed into the secondary structure. Secondary structure, there are two forms: beta pleated or alpha helical. It depends on the structure, right? They won't be asking what is beta pleated and alpha helical, but they have given example. They can ask the example. For alpha helical, the example is keratin protein. For beta pleated, the protein is spider silk fiber. This is an example. So just check this primary structure, secondary structure, and let me know if you have any doubts. If you are clear, we'll move to the tertiary and quaternary. Fine? Fine with these two? Fine in the sense, is it completely clear or is it manageable? You have some doubts, but it is manageable. Whether it is that the meaning? Or are you clear with all? Completely it's clear. Don't hesitate to ask the doubt. If you have any issues, if you can't understand anything, just ask me. I'll explain it. Right? If you can't understand, it's not your problem, it's my problem because I haven't explained it clearly. Right? So, do you want an explanation again? If you want, I'll explain again. No issue. Can draw the structure and I just explain it. Do you want an explanation again? So clear. If I make right. So, what should I say? These bonds, these intramolecular bonds that are formed between the same backbone of different amino acid. Same backbone components only, oxygen and hydrogen, right? So what you do is after this class leisurely, right, when you are free, just take and see the structure and compare with this top structure, how would it form, right? You should do like this: you read this and check the structure, read this and check the structure, then you will get used to it, right? Okay, let's move to the tertiary structure. Usually, the secondary structure polypeptide chain, which is formed alpha helical or beta paired structure, either form, they would further bend and fold. What happened during the secondary structure? They coiled and folded. Here, they are bending and they are folding extensively, forming precise, compact, unique, functional three dimension. These are the characteristic features of tertiary structure of protein. So underline them. If you have the dude, you can underline them. If you have the source book, you can underline them and highlight them. Right. So these are the characteristic features of tertiary structure. And during tertiary structure, what are the changes that take place in secondary structure? Secondary polypeptide chain undergoes bending and folding. Is that clear? Is that clear? Yes or no? Hello, are you there? Is it clear? This part? Not this complete part. I'm saying this until this dimension shape part. No, if you ask me like that, they won't be asking further out in this. They won't be asking what is alpha helical, explain the structure. No, they won't be asking like that. What they can ask is they will be asking secondary structure. What are the changes that can take place in the primary structure of a primary structure of a polypeptide chain to form into a secondary structure? What are the changes? It can coil and fold. Okay. They can ask what are the reasons for it to coil and fold. What is the reason? The intramolecular hydrogen bonds that are formed. Between what and what the hydrogen bonds are formed? We can say that it is between oxygen atom and hydrogen atom of the nitrogen atom of the same backbone polypeptide chain. Right. They can ask what are the two forms of secondary structure? Alpha helical, beta pleated. Examples of them: keratin, spiral cells. That's it. They can't ask further. That is, those are the questions they can ask maximum. That part not clear means what do you mean? You mean what is alpha helical and beta pleated? You are not clear with that part. Now, sir, no, sir, the next one. Next one means the tertiary part. The tertiary part. Yes. Okay. So this is fine, right? Primary structure, secondary structure, this is fine, right? You have any doubts? No. Okay. Then let's go to the tertiary part. Now, just think the secondary structure. At the end of the secondary structure, there is a secondary structure of polypeptide formed. Uh, what happens to that secondary structure of protein? Let's see. It will be bending and folding. It will bend and fold. Now, initially it was coiling and folding. Now here it starts bending. It bends and folds further, forming a precise, compact, functional, three-dimensional shape structure. Right? So there it took place due to intramolecular hydrogen bonds of backbone components. Here it is taking place due to interaction between what type of component? What type? This is taking place due to interactions between side chains R group. So simple. Can you understand? They are what happened. They are the primary in the secondary structure. What happened? The primary structure initial which was formed, it will be coiling and folding further due to intramolecular hydrogen bonds that took place between backbone components. Here, the secondary structure would be bending and folding due to the interaction between side chain groups. Is that clear? Is that clear? Now, if you have any doubt, just drop a message with clearly. I'll explain it. Do you have any doubts here? This part? Yeah, hydrophobic means it does not like water. It hates water. Hydrophilic means it loves water. Right? Part no, no that part. No. Which means now you are fine with this part. This is clear, right? No doubts with that. This passage is it clear? No doubts. Right. Now here they have given the type of interaction that can take place. The type of interaction that can take place between the side chain components. So they are what type of bonds were present between the oxygen atom and the hydrogen atom attached to the nitrogen atom? What was the bond that took place? I drew it in the yellow color. Can you remember? What was the bond? In secondary structure, only one type of bond was spoken. What is it? Can you remember? No, peptide bond was before that, during the amino acid formation. See here, what type of bond? Result of intramolecular. What type of bond? Intramolecular hydrogen bonds. Is that clear? So this one here, intramolecular hydrogen bonds, right? So here in these dot bonds, those are ones called intramolecular hydrogen bonds. So here only one type of bonds, they are intramolecular hydrogen bonds. So here during tertiary structure, there are four types of interactions that can take place between side chain. What are they? Hydrogen bonds can be present, disulfidic bonds can be present, ionic bonds can be present, van der Waal interaction or hydrophobic interactions can be present. Is that clear? Is that clear? Yes. So you might ask, sir, why this now there was only one. Here, why there are four options? Can you explain me? Why there are four options? Four types of interactions that can take place. Why? Just think. Why? Have any idea? Because in secondary structure, the bond was present between what component? Backbone or side chain? Backbone or side chain in secondary structure. Think carefully. Backbone. Very good. So backbone, does it change from amino acid to amino acid? Does backbone change? No, it is constant. Very good, very good. You have that. You have that. Fixed knowledge, right? Right. It is constant. So as it is constant, the type of bond is also constant. Only one type of bond, hydrogen bonds. Is it fine? Is it clear? Yes. Here in the tertiary structure, what type of bonds are present? What type of bond? In tertiary structure, the interaction of bonds are formed between what type of groups? Backbone or side chain? Side chain. Does side chain constant from amino acid to amino acid or does it change? Varies to each other. They are not constant. So as it changes from amino acid to amino acid, depending on the R group, these bonds also vary. Right? Depending on the R group, it can vary. Can you understand? Is that clear? It is not fixed. So there is no fixed type of bond that can form. Examples for tertiary structure of protein, most of the enzymes myoglobin, albumin. These are type of enzymes. Sorry, not these are enzymes and these are type of proteins where they are in tertiary structure. Is that clear? So this tertiary structure is slowly becoming functional. So compared to secondary structure has very very less functioning, but they have functioning. Some sort of function they have. So compared to secondary structure, tertiary structures are a bit more functional. Become primary structure does not have any functioning. So compared to primary structure, secondary structure have a bit more functions. Compared to secondary structure, tertiary structure have more functioning. Quaternary structure will be very very good functioning. So can you understand? They become slowly functional. Is that clear? Is that clear? Yes, right? Is it clear? Is it if it's clear, drop a message. Is it clear? Is this part clear? Tertiary structure. I checking with the note. Oh, is it clear? Right, very good. Then let's go to the last part, which is the quaternary structure of protein. Aggregation means collection, accumulation, or something, right? Aggregation of two or more polypeptide chains involved in the formation of one functional protein. To form one functional protein, two or more polypeptide chains can involve if it is in a quaternary structure. If a protein is in quaternary structure, how many polypeptide chains should be present? At minimum, two should be present, minimum. Two polypeptide chains should be present if there is a quaternary structure of protein produced. Is that clear? Is that clear? Clear. Separate chains are called protein subunits. Those chains separate, right? Now, two or more polypeptide means they are separate chains. Those separate chains are called protein subunits, right? Those chains are held in place by inter or intramolecular interactions. They haven't said what are those interactions. They have said inter or intramolecular interactions. Is that clear? Simply like this, made up of separate chains, polypeptide chains. They have said that minimum two. If you take two, they are separate chains, right? Each of them are called protein subunit, and between these, there are inter intermolecular interactions like this, or they can be intramolecular interactions that can be present. Intramolecular present like this. Is that clear? Inter means between two, intra means within the molecule. Is that clear? Is that clear? Do you have any doubts? No doubts, right? Okay. So this is clear. So examples they have given: quaternary structure, hemoglobin, collagen. So hemoglobin is present in our blood. In blood cells, they are present to carry oxygen. Collagen is present mainly in it is a one that is present in skin, right? It is useful in maintain. If you see in females, the collagen concentration of collagen is present in large amount so that their skin is moisturized and they are a bit fair and soft compared to men. So that is due to the collagen, right? So is this clear? Coordinate structure of protein. So simple, nothing much hard, right? Once if you just clearly go through them, you will find it that it is very easy. Is this clear? Coordinate structure of protein. Clear, right? Then shall I move to the next topic? So after this topic, a small table. After this table, I hope, yeah. After that table, here we will move with that table. We'll be ending protein today, right? Right. Okay. Denaturation of protein. Denaturation of protein means now every good story has a bad. There's uh, a bad incident also, right? So if there's a plain story, even in a film, in a movie, if the role play of the or the screenplay of the movie goes on like a constant line, the audience would not be feeling uh, an interest, right? So there would be something, a bad incident that would take place, a death of hero's a parent or his children or something like that, and they'll be increasing that hypeness, right? So like that, here there is no hype that is created, but still, here also it was a good story up to now. We were studying about their function, how the proteins are looking like, how they are arranged. Here they are saying what are the what are the effects that can change or alter protein, which means normally denaturation of protein means, let's see, now the definition itself says the actual idea. Denaturation of protein is the loss of specific chemical three-dimensional shape due to the alteration of weak chemical bonds and interaction. So it alters the shape, three-dimensional shape and nature of the protein. Is that clear? Why? Because due to the alteration in weak chemical bonds. Now we have learned here throughout this process there are large number of bonds. See here, there are four bonds here. Here also they have said intramolecular bonds. In the second structure they said that there are hydrogen bonds. So in these bonds, if there are any weak bonds, those weak bonds can lead to denaturation of protein. Or there is another chance. What? There are certain agents that can cause the denaturation of protein. They will be causing the denaturation of protein. Is that clear? What causes the weakening? These agents are the one that causes the weakening. Is that clear? These agents. They have given here below four agents. Those four agents are the one that are causing. Let's see what are the agents. High temperature, high energy radiation. High temperature, high energy radiation. Now, have you learned in your chemistry that if there is any energy that is supplied in excess, from the electrons start to escape and they'll be boosted to high energy levels? Have you studied in your chemistry? Have you started the chemistry parts? I don't know whether you have started. This is in your first unit, right? Have you studied that? When you supply energy in excess, electrons from their lower levels they get excited to high energy state. These high energy states are unstable, so they'll be getting reached back to initial locations. Right? These are given in your chemistry. So similar to that, here when the bond is present, when high energy comes here, that bond's strength weakens. Right? High temperature, high energy, they will weakening the bond. Right? Strong acid, alkaline, high concentration of salt. Now acidity, when it increases, also the bond gets weakened. Why? Because there's an alteration. Now normal pH should be maintained at a constant level. It should be maintained at a normal, healthy level. I mean the word healthy, it should be maintained at an accurate, beneficial manner. When it fluctuates into strong acid means 1, 2, that pH. When it is high alkaline, also it can lead to the weakening of bond. Heavy metals means metals like iron, arsenic, right? Molybdenum. Those are heavy metals. When those metals are getting exposed to proteins, they can lead to what? They can lead to weakening of this bond. And fourth one is organic solvent and detergent. Organic solvent means, we know the word organic. Solvent means a compound that can dissolve, a compound that lets other solutes to dissolve. You know the word solvent, right? From your oral knowledge. So those solvents and detergents can also make the protein weaken. Is that clear? This part, this complete part. H containing solutions. H2 carb. Organic, yeah. Organic means H2 contains solution, right? Carbon and hydrogen. So just check this denaturation of protein part and let me know if you are clear. If you have any doubt, no doubts. Is it clear? You mean you have doubt? You don't have any doubts? Just check it, right? So just check, no doubt. Right, okay, fine. So then we are coming to the end. We are coming to the functions of proteins. After this, we'll be completing the protein part. Right? Let's see. Type of protein. So depending on the function, these will be giving the function, the nature of protein, right? So nature. Catalytic protein. Catalytic protein means, example, enzymes. Normally, catalytic protein means they are acting as enzymes. They will be catalyzing the reactions. Their nature is that, right? Examples for catalytic protein: pepsin, amylase. What is the function of them? Catalyzes biochemical reaction. Is this first part clear to you? The type of protein, the example, and the function. Is that clear to you? Clear. Very good. Structural protein. Structural protein means they are contributing in structure wise. Right? Example, keratin. Keratin, think carefully. Keratin belongs to which structure of protein? We studied four structures: primary, secondary, tertiary, quaternary. Keratin belongs to which structure? Which structure? No, secondary, right? Secondary, wait, I see here. Secondary alpha helical or alpha helix, we can say both. Alpha helix, example, keratin, right? Okay. So function of keratin is prevents desiccation. So desiccation means loss of water, right? It prevents the loss of water, right? Keratin is present to prevent the loss of water that takes place, right? Second type of structural protein is collagen. Now tell me, collagen, it is what type of protein, which is what structure? So what structure does it belong to? Collagen. Primary, secondary, tertiary, quaternary. Let's see. Here's a quaternary structure example, collagen. So collagen belongs to tertiary, sorry, quaternary structure, right? So this is how they can ask, right? They can ask, they can give some protein and they can ask what is its structure, what is its function, what is its type of protein, right? So collagen, what is its function? It provides strength and support, right? Collagen provides strength and support. So this will be learned in the lessons of cell biology also. When we are coming more further with respect to plants, we will be studying that. Sorry, not with respect to plants. When we are coming with respect to the plant cells and animal cells, we'll be coming across the cell biology part, which means mitochondria, chloroplast, and organelles will be discussed. So there we'll be learning something called extracellular matrix, right? So there we'll be studying about collagens in detail. Here they have just said that. Now there will be studying how it contributes in strength and support. Here they have directly just said that collagens provide strength and support. Is that clear? These two, just check. Is it clear? Keratin and collagen. Is it clear? Yes or no? Is it clear? So keratin found in hairs and feathers. Yeah, very good. Keratin is found in hairs and feathers. So normally, if you see reptiles have keratinized scales. Keratinized scale, right? Why? Because they are preventing the water loss. They have keratinized scales. They'll be preventing the water loss, right? So keratin is found in hairs. Why? To prevent the moisture loss. It should have, now if you see in our hair also, those hair, the head is a part where it gets more getting exposed to sun rays, high energy, temperature, right? If there is no water, the moisture content, if it decreases, they start to dry. So the scalps get dried, right? So similarly for the plants also they have feathers which should be keratinized to prevent that water loss. Why? We have to moderate the temperature. We should maintain the temperature. So that temperature is maintained by this keratin. How? By preventing desiccation. Can you understand now? How its contribution. Is that clear? Yes, very good. So let's go to the next part. Storage proteins. Example, ovalbumin. This stores protein in egg. Now, egg, we know that egg is a source of protein. Normally, gym freaks. We used to say the individuals who are involving in gyms. They mainly have a very good aspect with respect to eggs. Why? Because mainly their routine goes on with egg because they have guided that saying that the egg is a good source of protein. So they'll be consuming eggs in large quantities, right? But actually that egg has protein. Why? Just because of this ovalbumin is a protein. Ovalbumin is a protein that is the one that causes, just because eggs contain this ovalbumin, they store the protein in them, so they are giving the eggs a protein nature. Can you understand? They are the one that stores protein in milk. Sorry, stores protein in egg. Is that clear? Ovalbumin, storage of protein in egg. They are the one that stores the protein nature in egg. Is that clear? Just because ovalbumin is present, eggs get a protein nature. Is that clear? Is that clear? Is it clear? Is that clear? So I can move. Yes, right? Okay, very good. Casein. Casein stores protein in milk. Now we know milk is also a source of protein. Why? If you ask that question normally they can't answer. Now from small days onwards we used to say that protein is egg is a source of protein, milk is a source of protein. But if you ask how, why, they can't answer. Now we know as bio students why. Egg is a source of protein because it contains ovalbumin. Milk is a source of protein because it contains casein. Is that clear? So these protein, what is their function? They store protein in them. They store protein in them, so making their eggs and milk uh, protein in nature, becoming them as protein sources. Is that clear? The storage part, is that clear? Yes, very good. Then let's move to the transport. Some proteins are involved in transportation function. Example, hemoglobin. It transports oxygen, carbon dioxide. So normally red blood cells contains hemoglobin in them. Hemoglobin will be carrying oxygen molecules. One hemoglobin molecule can carry four oxygen molecules. So it will be carrying oxygen molecules within the blood through various organs and tissues. Right? So it is acting as a transport molecule, transport protein molecule, so that it can transport oxygen and carbon dioxide through blood. So tell me, hemoglobin belongs to which structure of protein? Primary, secondary, tertiary, or quaternary? Check here. Quaternary. Right? Okay. Let's move to the other example for transport proteins. Serum albumin. What they do? They transport fatty acids inside the body. Serum albumin transports fatty acids. Right? Is that clear? This transport parts so small functions, not even one complete statement, small ones, right? They can ask them, right? Is that clear? This transport part. Yes. So we can move to the next part. Only I guess yeah, we have only three more, right? Right. Hormones. So some proteins act as hormones also. So they can ask what are the main natures or characteristic features of protein. Means they can ask now if they ask what are the type of functions they can render. We can say they can act as catalytic proteins, they can act as structure protein, they can act as storage protein, we can say they can act as transport proteins, as hormones, contractile or motor proteins, defensive. These are the some main types of proteins they can involve, right? So hormones, example, insulin, glucagon. Do you know insulin? Normally, if you have any, if you're, if you know anyone who is affected with diabetics, right? They know what is this insulin means, right? Normally, diabetic patients they will be given with insulin injection doses, so they should insert them, right? Why? Because insulin reduces the blood glucose level, right? Glucagon increases the blood glucose level. Now, don't think glucose does a bad thing, right? No. When the glucose level in the blood decreases than the normal level, glucagon will be increasing its glucose level, regulating. So insulin glucagon involves in regulating the glucose levels. Is that clear? Is that clear? Yes. So what does insulin do? Insulin decreases the blood glucose level when the blood glucose level is in excess. Now, when we eat more and more sugar type of compounds, when we eat more and more glucose containing compounds, which is normal carbohydrate compounds, if we consider large in concentrations, what happens? The body's glucose level increases. So, in our human body, normally insulin and glucagon are present. In our human body, insulin and glucagon are present, right? So they will be decreasing and altering those. If it is in excess, insulin is secreted, insulin will be decreasing its level. So when it is in very low level, when the blood glucose is very low level, glucagon comes and gives an upliftment, it increases it. So for the diabetic patients, why they give an insulin injection or doses? In diabetic patient, this insulin secretion is comparatively low, so they give externally some doses. So right, is that clear? So these are not needed for you. These will be discussed in the later part of your lessons, right? Is that clear? The hormone part. Hormone part. Is that clear? Yes or no? Clear, right? Okay. Let's move to the next part, which is contractile motor, right? Example, actin myosin protein involves in contraction of muscle fibers. This protein is the one that contributes in contraction of muscle fibers. Example, skeletal muscles. Skeletal muscles contract and relax due to these proteins, actin myosin. How? We'll be learning it in unit number 5. Now in your unit number 2, you are in unit number 2. Unit number 5 is completely about human body. So they are under skeletal system. We'll be learning how actin myosin contributes. Now for the time being, no need to learn about them. Is that clear? Now we should know that under contractile motor nature, actin myosin is present. They involve in contraction of muscle fibers. They involve contraction of muscle fibers. Is that clear? Is it clear? Is that clear? Yes or no? Do you have any doubts? So the structural variation occur. Structure variation means depending on the structural components, the function varies. You have asked it in opposite manner, right? Depending on the structure, their function varies. If you see secondary structure has keratin. Keratin, what they do? Keratin was in preventing desiccation, right? If you see tertiary structure has immunoglobulin, right? Immunoglobulin. Now you see, next one is immunoglobulin. So immunoglobulin belongs to that part. So it has another function. It involves in eliminating foreign bodies, right? And if you come to quaternary, collagen, hemoglobin. Collagen involves in providing strength and support, right? Hemoglobin involves in transportation of oxygen. So depending on the structure, their function differs. Is that clear? Is that clear? Shall I move to the next part, the defensive part? Okay. Defensive nature. Example, immunoglobulin. Immunoglobulin, right? So immunoglobulin, simply they are called antibodies, right? So these immunoglobulins, what they do, they are eliminated foreign bodies when the body is affected with some sort of virus, some sort of disease. These immunoglobulins are the ones that are fighting against them, right? Is that clear? Antibodies, they are the ones that are fighting against them, eliminates foreign bodies, right? Simple as that. So that's the end of protein section. Is that clear? Did you have any doubts throughout this lesson part? So next class, hopefully we'll start nucleic acids. And if we complete this, the first tute will be done. No doubts, right? Everything is clear. All clear, right? So make sure to revise this complete section until protein. So hopefully today or tomorrow you'll get an essay, right? Based on this, I'll give one essay. So weekly, I'm planning to give only two essays for you. Just you try them, right? And you can send the photograph of the essay written, or you can, rather than sending the scheme, if you send me the picture of the essay you have written, I can mark it and send you, right? So that would be preferable. And I'm planning to give a MCQ paper every once in a 2 weeks, right? And make you in touch of the content, right? Okay, then that's it for the day. Thanks for joining live, and it was a great help being throughout this session, right? So thank you. So let's meet. Hope to meet you in a next session, which will be on next week, Friday at the same time, right? Okay, then. Thank you. Goodbye. Welcome.